When you think of a chromosome, you likely picture the microscopic X shape found in a dividing cell. But certain specific tissues produce giant, oversized variants. For your Class 12 biology board exams, you need to understand these special chromosomes through three distinct criteria: where they are found, how they are built, and what they do. The standard tightly packed X chromosome is built for safe transport during cell division, not for active reading. Giant chromosomes exist because the cell requires an immediate massive surge in RNA and protein production. To meet this structural demand, organisms rely on two main types of special chromosomes: polytene and Lampbrush. Unlike typical chromosomes that are visible mostly when the cell is dividing, these giants remain visible during the working, active phases of the cell cycle. We begin with polytene chromosomes. They were first discovered by E.G. Balbiani in 1881 in the salivary glands of fruit fly or Drosophila larvae. They reach their enormous size through a mechanism called endoreplication. During the S phase, the DNA replicates over and over again, but the sister chromatids fail to separate during anaphase. Instead, over 1,000 identical strands remain bundled together at a single centromere, forming a thick cable. If you look closely at this cable, you see a distinct pattern of alternating segments. The dark bands contain highly condensed, mostly inactive DNA, separated by lighter interbands where the chromatin is more relaxed. When the cell needs to read a specific gene, sections of these dark bands uncoil and balloon outward. These expanded areas are called chromosomal puffs or Balbiani rings. This unwinding exposes the DNA, allowing enzymes to rapidly transcribe it into RNA. By having over 1,000 copies of the exact same gene lined up and transcribing at once, the fly larva can manufacture massive quantities of salivary glue proteins needed right before it pupates. The second type is the Lampbrush chromosome. These are the absolute largest chromosomes known, found primarily in the growing oocytes of vertebrates like amphibians and birds, but they are notably absent in mammals. Pay close attention to their timing. Lampbrush chromosomes are distinctly visible during the prolonged diplotin stage of meiotic prophase 1. This micrograph shows their core anatomy. Tightly condensed chromomeres form a central axis. Filamentous loops extend outward, resembling a lampbrush. Each loop is an area of hyperactive RNA synthesis. The DNA unspools, granting transcription machinery maximum access. The oocyte relies on this rapid, high-volume transcription to synthesize the enormous amounts of yolk and lipids that an embryo will need to survive its early development. While the polytene strategy multiplies the number of DNA strands thousands of times, the Lampbrush strategy uses just one pair of chromosomes, maximizing their accessibility through radical, extended unspooling. Let's test your active recall. Take 5 seconds. Name the specific tissue source and the cell cycle stage for both of these giant chromosomes. Let's check your answers against this comparison table. Polytene occurs in insect salivary glands during interphase. Lampbrush occurs in vertebrate oocytes during the meiotic diplotin stage. Structurally, polytene forms a multi-stranded cable with puffs, while Lampbrush has a condensed axis with lateral loops. They look entirely different under a microscope, but both structures solve the same biological problem. When a cell must produce vast quantities of protein and RNA at an accelerated pace, it builds giant chromosomes to complete the task.